So in the last session, I have told you regarding how to use Notepad for saving the Python file. I have done certain things. For example, I have declared a variable d and f, and I have multiplied these things and save in a variable that is s. And I want to print that value. And so because it's a number value, we have to convert this number into a string variable. So I use that thing, and I'm using just a plus thing for concatenating two strings. Concatenation means addition of two things. We can add two things simultaneously. So one is string and another is also converted string. So how can we see the result? Here no run button is coming. So that's why Notepad is not recommended for writing this thing. You can write that, but after that you have to save that thing in a py format. So that you have to go towards this thing. What is the name of that file which I've saved here? For example, cal. So it is there. So in this case, for example, I'm opening it with or editing with it ideally. I have to again open it with that, and after that here run option coming. Here it is. So I'm running that thing and it is showing me the result of multiplication. You can use either way. Either you can write the scripts in a Notepad file and after that run it in ideally format, or simply use ideally format here, make a new file and do all the calculation, all the things here, all the scripting kind of thing here, and save that file and run that file also. So it's depending upon you. I will recommend that you should use ideally platform here. So for making your scripts in an editor mode, for example, here you are making and after that run that thing, because in your assignment you have to submit two things. One is your script. Either you can submit your script in a Notepad format, you are just copy pasting that thing in your Word document, and you have to take the screenshot. For example, right now I am using this ideally platform. I am showing you why it is important, because when you take your FASTA sequence, you have downloaded your gene sequence in your system. I think everyone has done that. This FASTA file, we have to read that. If we are reading that from the Notepad file or from the FASTA file which is available on your desktop or any other location, it is difficult to fetch that. Instead of that, you have to go towards the installation part where it has been actually installed. For example, in my case, this my PC is there, C drive is there, user is there, and in this user, I am to use my name of the PC is Dell. So it is here it is app data. You have to go for the app data, then go for local. Choose top. These are programs. In this Python is there, Python 39, because my version of Python is 3.9, so it is Python 3.9. So all the things which I am currently working, it will save here. I think I've showed you a script regarding cal in this case, where I have used ideally editor. In this case of ideally editor, when you open this thing, the script will be open. So it was there, A is 34, B is 35, and we have used that thing in a third variable, that is C, and we have printed that string in which the value is given. For example, in C, the value is already allocated. So I will recommend you to use this ideally platform for your FASTA sequence or your other gene sequence. So how can we do that? First, we have to download our gene sequence. For example, I have already downloaded that. Rather than saving it in a normal format, you have to change the format. For example, here it is NCBI. Select your gene sequence. Your accession number is already given to you, but I am choosing any random gene. For example, I'm going to the nucleotide sequence. Here it is. I'm using insulin. I'm using any random FASTA sequence so that I can demonstrate you how you have to save your file, FASTA file. So, for example, I am using Octodon Digus insulin sequence. Here it is FASTA format. I'm using that, and I'm just sending it to file format. The same experiment which I have done in the first FASTA format, it is downloaded here. It is showing sequence.fasta. I'm opening it. So for example, I'm opening it here. Open with, for example, Notepad I'm showing. Here it is. The format of this particular file is .fasta format. You have to use this .fasta file by converting it. For example, here it is. Here it is .fasta already there. If you are just copy pasting it into a Notepad file, you have to use a format that is .fasta so that it can recognize your format. So I'm using this thing and copy this sequence file. Here it is. Go towards the location which I have told you. Here it is Python 39 because my version is 3.9 and paste here. So here it is. in my case, I have already pasted my DNA sequence here. So here my DNA sequence, for example, insulin, Homo sapien insulin is there, and the gene name is IRS1 gene is there, mRNA sequence. As I earlier told you, in case of bioinformatics, we cannot use mRNA AUGC. We are using cDNA sequence, alternate of that. So I'm using that only. So in case of this Python, we will use different kind of methods or different kind of variables. The most common one, for example, I have told you how to declare variables. For example, there is a variable known as DNA. In earlier, I have told you how to use command prompt. This is command prompt. When you click on new, then it becomes editor mode. If you want to simply run certain small commands, then you can use simply this command mode. Otherwise, if you have to write certain bigger scripts, then you have to go for editor mode. For example, in the earlier session, I have told you about how to declare certain numbers, A, B, C. We have done that calculation. We have done addition, subtraction, multiplication, division. Right now, we are defining certain strings, because in case of bioinformatics, we are working on DNA, RNA, and proteins. And I have told you in case of DNA, we are working on ATGC. In case of mRNA, we are not considering AUGC, rather than we are considering cDNA. In case of amino acid, we are not considering B, J, O, U, X, Z. Other letters are considered as amino acid sequence. For example, I am defining a DNA sequence A, T, G, G, C, 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 T, G. This thing is there. One thing you have to clearly see that you have to use certain quotes, either double quotes or single quotes, for defining a string. If you are defining a string, you have to use either double quotes or single quotes, but use it in a balanced manner. For example, no one can use like this, sir, can I use this? A single quote is there, other is double quote. If you are using initially, you have to use again single quote. I'm showing you how it is actually showing you the result. I want to print this DNA sequence, so print. I want to print which DNA sequence is there. You do not have to use double quote while printing this thing. For example, I have to first save this thing. It is showing which name. For example, I'm using testing. Name is testing. So here it is. The sequence has been printed. The sequence which you have written here, I think this should be closed. Here it is. The sequence which you have written here, when you click on run module, it will run automatically. So print command is very useful. Also, this language is quite case sensitive. You cannot use any, for example, if you are using capital P here, print is there, it will not run. It will give you error. For example, I'm showing you, it will give you error. So these keywords are quite important, and they are quite case sensitive also. Also, in case of this DNA, I have told you we have to use quotes. Either you have to use double quotes or single quotes. It will not, you can say, damage your results. For example, right now, in the earlier example, I've used single quotes. I'm using double quotes right now. So is it giving any difference? No, no difference is there. But the only thing is that you have to make it as in a balanced way. Also, we can use certain keywords. For example, have you heard about this new line character or tab, anything in the case of C or C++? Yes, we can use also in this case. For example, here it is. Instead of this DNA, I am using, for example, here /n, and after that again A, T, T, G, G, C, C, C is there. So I'm using this thing and running this thing. So here it is. First sequence is there. Due to the /n thing, it comes in the next line character or new line character is there. Other than that, we can also use /t is there. So you can use any operator in this case. So it is same as that of other languages which you have studied. But the good thing about this is that it is not that much laborious. You have to simply type the keyword or type the operator, and it will show you the result. The other thing I want to mention is that we can use Python for the concatenation thing. For example, two DNA sequence. Have you heard about this exon intron thing in your genome sequence? So for example, I'm saying there is an exon region. Exon 1 is there. Also, as I earlier told you, it is a case sensitive language. If you are writing this capital E, so you have to remember while writing this printing command that it is in capital. Right now I have converted into a small one, that is another exon is there. Whose sequence is this? For example, GGG, CCC, TT, anything is there, any combination is there. So this one is there. So I have to combine these two. I have to join these two. How can I do that? I am using, for example, I'm using RNA. another variable is there. So I'm adding this thing. I can add two common thing at a time. For example, if I am adding two strings, I can do that. If I'm adding number and a string, I cannot do that. For converting this thing, I have to either convert my string into an integer or integer into a string, as I earlier shown you the example. So in this case, for example, x1 as well as x2 is there. Here it is, and whole result is printing under RNA. So I want to print what is the exon, what is the exon 2, and what is the combined sequence. So here it is. I'm writing the exon 1 is here. The thing which you have to mention is either in the double quotes or in the single quotes, or the thing which you have to print, which thing I have to print, it is exon 1, which is a kind of string. Here it is a string. I'm also printing the string. So I have to just write exon 1. Rather than I have to engulf them into a string operator. I do not have to do because it is already a string. After that, so I have to run this thing. For example, I'm seeing it is able to run that thing. So yes, the exon is this one. So there is no space. So I have to put that space also, or you can use /t operator here also. So here it is. So it will show you that I'm just copy pasting that thing so that it will take less time to do that. It is exon 2. I'm converting it here as exon 2. So it will print exon 1. After that, exon 2. Right now I have to print the result also. So the resultant RNA sequence, the resultant RNA sequence, write anything which you want to print. So I'm using that thing is I have to write just RNA. So it should print like this. So it is asking for saving. So exon 1 is this, exon 2 is this, and the concatenation thing is also there. So we can concatenate the string by simply using a symbol that is known as plus symbol. So also we can find out the length of that particular sequence also. I'm using that thing for the length command. Here it is. I want to find the length of exon 1. So how can we do that? So here it is. There is a command that is known as len command. So len of exon 1. There is a variable that is known as len exon 1, in which the length of exon 1 is printed. So it is exon 1. Here it is. Same, I have to find out the length of exon 2. So I'm just changing this particular variable. This is exon 2, and I have to print this length into the print command. Can anybody tell me what is the answer of this? Not exact number, but what will be the variable? What is the thing which come out from this len exon 1 thing? Because in case of print, we have already written certain scripts. So I have to convert this particular thing into a string variable. So I'm using the length. Here I'm writing the length of. Okay, I'm using the same thing. Just copy pasting that thing so that I can get whole idea. So here I'm putting the length of exon 1 is, I'm using another variable. Here it is len exon 1. So I'm just putting it here rather than exon 1. I'm putting here it as length of exon 1, but it is a number numerical variable. So I have to engulf that it into a string variable. So here it is. I'm using str string. So right now, whatever number it will came, it will add towards this. Same thing I have to do for the exon 2 also. So here it is. I have to just convert it into 2, and here it is 2. Also, I have to find out the total length of the RNA. So here the other command we can write here. For example, the total length of RNA is, so I didn't define any variable, so it will generate an error before that. So I have to put this here. So len of RNA, which is equal to len of which variable? RNA variable. So RNA is there. So here it is. So all the values of length of RNA will go towards this len RNA variable. So I have to write here, that is len RNA. So all the things has been written here. So I have to just save that thing and run that thing. So it is quite clear. The exon 1 was this and its length is this one. Exon 2 was this, length is this one. Exon 3 is this and the length is this. So you are doing some different things simultaneously. You are adding the things, you are using a strings also, you are concatenating that thing. All the things are going simultaneously. So this is the power of Python. You didn't do anything new. You are just using a simple command kind of thing. That's why it is known as higher level language. You are using common keywords which you are using in your normal language. So I have done this thing. Also, there is an important role of comments. Have you heard about this thing? Here it is, for example. Here, whenever we have to write certain comments, we have to use hashtag. So it is quite simple. It's common to that of other language also. For example, this program is for splicing. Okay, this kind of thing is there. You can add, for example, other things are there. For example, here it is useful for the other programmer also to read your code if comments are there. For example, this is for finding the length. So this thing is there. So we can use these comments kind of thing in the scripts also. So whenever you are writing certain codes, please use comments so that I can able to read that thing in a user-friendly manner. Otherwise, it will become quite difficult for me to read your entire code because everyone has its own variable name. For example, here I have used exon1. Someone can use, for example, some shorter form ex1 he can use. So anyone can use any kind of variables. For defining those things, we can use comments. In case of arrays also, can anyone tell me what is array? In case of Python, it's a collection of any kind of data. For example, in first, for example, it starts from 0 and 1, 2, 3 and so on. For example, up to n it is there. So I am showing here 4. So what is the length of this array? How many variables are there in case of this array? 5. So the first location is 1 and the last location is 4. This thing you have to remember. For example, I'm using a common word. For example, okay, I'm using a DNA sequence A, T, G, C, C, anything is there. Can anybody tell me what is the length of this DNA sequence? This is 3, 6, 9. What is the maximum position of this particular DNA sequence? 8, because it is starting from here 1, 2, 3, 4, 5, 6, 7, 8. Here it is there. So if I'm, for example, saying it is a variable known as DNA. In case of array, we can use DNA and we have to specify the position. For example, I am talking about 7th position. Can anybody tell me what is the answer? What is DNA of 7th? G. Here it is. So it is there. For example, I am saying here at DNA of -3. What will be the answer? In Python, it will run because it assigns the reverse order also. For example, here it is. So the third value should be nothing but the G. For example, I am talking about -8. So it is -1, -4, -5, -6, -7, -8. So the -8 is nothing but T. What is the value of this? DNA -22. The position is not available. So I'm showing you this how it is possible. So I'm using command prompt of Python. So here it is. DNA has been defined. The only disadvantage of this thing is that using command prompt it is that it is not saving your all the variables. All the data is lost when you close this thing. So here it is A T G C C C T G G is there. DNA has been defined. The variable has been defined. I have to find DNA of, for example, 6th position. It is G. For example, DNA of -6 position. I am talking about this. It is showing me this. For example, I am showing DNA of -22. It is showing me an error because it is out of bound. It is not showing you the exact value. So it is quite specific. It is quite, you can say, robust in terms of running the commands. So in case of bioinformatics, we are using arrays and we will find out the location. Can anybody tell me what is the role of location in terms of DNA? Why it is useful? Why this location or the arrays kind of thing is useful in Python or in bioinformatics specifically? Talk about the arrays kind of thing. How can we find out the location of that particular thing? Right now, I'm going towards one step further. Here it is. I've told you regarding this, how to define a DNA, how to print a DNA sequence. Also, I have told you how to concatenate a DNA sequence. I have also told you how to find out the length of the DNA sequence. Also, how to print two different kind of variables. For example, here it is string and here it is a number. So we have to convert a number into a string. How can we do that? Also, the next thing is how can we lower down? For example, right now I am using this thing in a capital manner. How can I convert this uppercase into lowercase? We can simply use a command that is known as lower. For example, my DNA is ATGC and I'm using a command that is known as lower command. It will convert entire DNA sequence, entire this variable into small thing. I'm showing you how it can be done. Here it is. My DNA is there. I'm converting my variable, this variable, into lower by simply writing dot lower. It will automatically convert. Okay, save. For example, T is there. I'm using save it there and running. It is not showing IS, it is showing the result. So it has been converted ATGC. So we can use lower command to convert the upper variable into lower variable. My question is, if my DNA sequence consists of both, for example, accidentally I got a data, for example, DNA is there, which consists of A, C, T, G, A, C, this kind of thing is there. Can this lower command will work on this or not? Yes. For doing this thing, for example, sometimes you can use upper command also, dot upper, for converting this randomly organized upper and lower thing into a common sequence. For example, you want all in capitals, so you can use upper command, or if you want all in small, then you can use lower command. So this is quite useful. Another command is useful, that is I have talked to you about this concatenation kind of thing. I've already told you how to add this thing, concatenation thing. These are things. Another thing is that is count command. So I'm converting this. For example, a protein sequence is there. I have earlier told you in case of protein, DJOUXZ we didn't consider in bioinformatics. So this is a chain of amino acid. We have to count the amino acid. So first I have to count a amino acid that is known as valine. So I'm using a command that is known as count command. In case of this variable protein, I am just adding dot count, and we have to find which particular string variable, that is not point, shift only V. So if V is present, then it will automatically counted and it will store in this variable that is known as valine count. For example, you have to count a triplet protein amino acid, or you can say peptide sequence, LSP, whether it is present or not in that particular, you can use LSP and it will automatically give results in LSP count. And again you have to count for example, amino acid that is known as tryptophan, W. So whether tryptophan is available or not, it will automatically count and it will return this whole number in this particular variable, that is tryptophan count. How can we print that? Simply, for example, I want to print valines. So valines are nothing but valine count. So it is a number. I have to convert that into a string. So this is a string and it will show you the result. So I'm showing you how it is there. So in case of this particular sequence, 2 are valines, one pattern of LSP is found, and this tryptophans in that particular amino acid is 0. So I'm using this same command for DNA sequence, and I'm curious to find out whether it consists of any start codon or not. For example, I'm saying here ATG. I'm using a random thing. Okay, this is a sequence, so I'm converting it here. For example, DNA, I'm asking, here it is DNA. I want to calculate start codon, for example. So it is START codons. So I have to calculate start codon. My variable name has been changed by DNA. So DNA, I have to count DNA from DNA, which kind of thing? What is start codon? Can anybody tell me? ATG. So we have to use ATG. Please, I am again asking the same thing again and again. It is very case sensitive language. You cannot use ATG here because in the string it is all in capitals. So you have to use ATG. That's why I am saying the sequence which you have downloaded from NCBI, please check that whether it is in uppercase or lowercase. Either convert it in uppercase or use uppercase command, or convert it in lowercase or use lowercase command. So this is start codon. Can anybody tell me what is stop codon? How many stop codons are there in case of this codon table? 3. first thing I have to do, first I have to calculate the start codon, how many number of start codons are there and how many number of stop codons are there. I by mistakenly I have used a capital thing, so I am converting it in lower so that it will not giving an error. Also, I want to find out number of A. So, for example, A count. I have to count number of A's that are present in this particular sequence. So here it is. Again, I have to count D and A count. Which thing I have to count? A. Also, at the same time, I want to count T, number of T's that are present. So I'm using here T, and here I'm using this capital T. I want to find number of start codons, number of stop codons, number of A, number of T. So I want to print the values. So here I want to write the number of start codons are, for example, how many? What should I write here? Start underscore codon. Why I have used this str? Because it's a number. I have to convert into a string. These common things are quite useful while writing the command. Otherwise, it will generate an error. Also, at the same time, I want to find out the number of A and T. For example, the number of A. So I'm using here A's. The number of A's are, for example, here the answer should be A underscore count count. So it is there. It will give you number of A's. Also, I want to find number of T's. So here it is T, and here I am using just T. So in this case, should it print all the results? My question is that. All things are correct. Should it print the results or not? We are counting what? Start and stop. Stop and start. So here it is. Now print the counts. It is correct. So right now I am running this thing. So here it is. Number of start codons is 4. Number of stop codon I didn't print. So I have to again go towards this, and I have to just write number of stop codons if it is present. It is stop. I have to just save it again, and this one. It will give you number of start codons, number of stop codon. No stop codon is present. Number of A's and number of T's. Can anybody tell me what is the formula for calculating number of AT in the DNA sequence? Number of A plus number of T upon means A plus T plus G plus C. This kind of thing into 100. Can we shorten this thing by using Python? We have counted this. Count A. We have done that. Plus so here it is A. After count T we have done. After that we have calculated the length. The length of DNA sequence we have calculated, and if you multiply it by 100, it will give me the AT percentage. Are you getting my point? Import your sequence, FASTA sequence. I'm showing you that in this case. You can take a screenshot regarding that. One command that is useful for reading out the DNA FASTA file. Please, I am saying again, please save your file at this location, which I have told you, Python 39. Otherwise, if you are saving on the desktop, it will not read out your file. So I have saved my file here. For example, I'm showing you here. This is my FASTA file. I've shown here, it is Python 39, where I have saved that thing. After that, there is a command that is known as open, which will read your DNA sequence. After that, we have to remove the first line, because first line starts with the greater than sign, and certain little bit on information is there. We have to remove that. So read line is there. That will remove first part. We have to consider the rest sequence part. So my file, the name of my file is my file, or the variable of my this thing is my file. So I'm reading that thing. It will read all the sequence information, and it is replacing the gaps which is present in your FASTA file. If no space is there, if next line character is there, it will remove that thing and it will make a continuous string of your DNA sequence. And this command is useful for, can anybody tell me, length? Other, we have to print that particular thing. Number of characters are there, and it will print the gene. So I have run this thing. So this is my gene sequence, because it is quite huge. It is showing here 123 lines are there. I have also find out the reverse sequence here. So number of codons are there, number of stop codons are there, A, T, and what is the length of the sequence, and what is the AT percentage. You have to calculate the GC percentage. Also, you have to find out A, T, G, and C. and number of start codons and stop codons.